Hi, my name is Jeff Marks, and today I'm going to be speaking to you about bone grafting options and, most importantly, the evidence available on each of these bone grafting options to support their choice for your use in your clinical practice. For today's discussion, I'm going to start off with an introduction to Cerapedics. I'm then going to walk through each of the bone grafting categories as they're available, which are Autograft, Allograft, DBM, BMP, synthetic bone grafts, stem cell products, and then I-factor peptide-enhanced bone graft. I'm going to conclude the discussion with a review of the published clinical evidence that's available to support each of these categories in your clinical practice. Cerapedics is a privately held medical device company located in Westminster, Colorado, focused on the development and commercialization of peptide-enhanced bone grafting technologies. The core technology, P15 peptide, was first brought into clinical use in the field of dentistry in the 1990s by Cerapedics' predecessor company, Ceramed. Cerapedics brought these applications into spine and orthopedics in 2008 with the CE mark outside of the United States, and then in November of 2015 with achievement and receipt of an FDA PMA approval for use in anterior cervical and discectomy and fusion. This PMA approval makes this P15 peptide enhanced technology only the second FDA approved drug device combination product for use in spine and creates an entirely new category of bone grafting materials, peptide-enhanced bone grafts. On the basis of the IDE study, P15 peptide showed a clinical superiority to Autograft in overall outcomes, had a tremendous safety profile, and is the only commercially available bone grafting product that will not put hospitals' reimbursement at risk for use in anterior cervical discectomy procedures. The bone grafting market as exists in the United States today is a very large one, representing over $1.5 billion in revenue. And it's comprised of a number of different categories of products that have varying levels of evidence and science to support their use. These categories, as we're going to talk about them today, are Autograft, Allograft, DBM, synthetics, the stem cells, and we're now going to introduce a new category, peptide-enhanced bone graft. I think it's important to spend some time talking about the regulatory process and pathway by which these products come to market and become available for your clinical use. I think there's a lot of misunderstandings, and I think talking about the regulatory pathway will help us understand in a minute why there's such enormous variability in the evidence, both clinical and preclinical, available to support each of these choices. At the top of the pyramid, the most challenging regulatory pathway belongs to the products that are drug-device combinations. So the FDA reviews these under a pathway which requires an IDE, investigational device exemption, and a PMA, pre-market approval. The second tier category of products really represents the largest in terms of number of products available in the market, and these are the 510K products. So with the introduction of the Food and Drug Act of 1976, the FDA recognized the large number of products that were currently commercially on the market were effectively grandfathered in. Any new products that were to be brought to market, if you could demonstrate they were substantially equivalent to these grandfathered products, you could file a document called a 510K, and upon successful review, those would be cleared by the FDA and could be brought into commercial use. Now, typically, that only requires an animal study, or in some instances, two rather small animal studies to support those filings. The next category, which are the human cell and tissue products, HCTPs, or minimally manipulated homologous tissue. These category of products really have no pre-market review for safety or efficacy by the FDA, but rather the companies manufacturing them have to follow some published guidelines for the proper processing, procurement, documentation, etc. And so these are typically brought into commercial use without any evidence, in many instances, whether that's animal or human. As we discussed, and largely a factor of the regulatory processes, the ranges of evidence that are required to bring these variety of products to market is enormously varied. Most of the companies commercializing these products really don't want to spend much more money above and beyond what's required to get into the market. Rather, they'd prefer to spend their money on marketing, sales reps, commissions, etc. So once again, at the top of the pyramid, the class 3 products requiring an IDE and PMA, those will always have a prospective randomized level 1 clinical study. In the majority of instances, those will be published upon their completion. So you have very solid clinical evidence on which to base a decision to understand both the safety and the efficacy of these products, as those are the FDA's requirements. In the Class 2 or 510K products, the FDA's threshold is really more about safety and less about demonstrated efficacy, although they do require at least one animal study. Today in spine, the FDA's requirements for a 510K are a posterolateral fusion in a rabbit model, but historically many products have gotten approved and used in the market on the basis of something as simple as a drill defect in a rabbit femur. Now, if you look at the labeling on these variety of products, you will see some of them are labeled for extremities and pelvis. That typically uses that drill defect model. Those that say spine most typically use the rabbit model with the posterolateral fusion. And as we had discussed previously in the regulatory pathway, the HCTP or tissue products, allograft tissue, or the homologous stem cell tissue products, those typically have no required evidence prior to their submission to the FDA, really just demonstration of their handling properties such to remain safety, try to remain viral inactivation, etc. So typically there are little to no documented evidence in either animal studies or human clinical experience unless physicians choose to do those on their own outside of the auspices of the companies that are producing and marketing these products. I think the first and most important category to discuss in any good bone grafting talk always is autograft. Now, autograft, and in particular iliac crest cancellous bone autograft, has remained our gold standard for performance for many decades. I think there's very little argument to be made that anything to date has been demonstrated to be better than autograft. And if there were just a magic kidney basin in your operating room full of iliac crest autograft, this entire $1.5 billion industry would have never had any reason to be developed. So why is it that autograft performs as well as it does? I think it's because simply it provides all three of the biological functions needed for bone formation, in that it is osteoconductive, osteoinductive, and osteogenic. While I won't be using those terms much through the rest of the talk, I think it's worth defining them fairly quickly. Osteoconduction is the property of providing a surface which is chemically appropriate such that bone can lay down directly onto its surface. Osteoinduction is the property of providing signaling molecules to turn on the native process for bone formation. And then osteogenesis is the property of providing living cells which themselves do the work of laying down the bone. Most typically, these will be mesenchymal stem cells or daughter cells of the MSC lineage, leading down to the end of that lineage for bone formation, which are osteoblasts, the cells that actually do the work of laying down the bone. So in addition to being our gold standard for performance, autograft is also safe. There's no donor-host rejection potential because it is the patient's own tissue, and there is no potential for disease transmission, again, by virtue of it being the patient's own tissue. While autograft has been our gold standard for performance, allograft has been a second-tier choice for quite some time, with a couple of good reasons. The first of which is allograft tends to be the least expensive option available in most hospitals. It's also been shown to be clinically efficacious, in some low-demand applications such as adolescent scoliosis, certain trauma applications with a good bone bed, and as an autograft extender where you don't get enough autograft in a particular patient. Now, with those advantages come some significant disadvantages. Because allograft is human tissue, it carries a potential for disease transmission. Now, while the processes for reducing those disease transmission potentials have improved over the course of years, that potential can never be eliminated, can never be brought to be zero. Now, along as we've discussed, the regulatory pathway for allograft tissue is guidance documents. There is no pre-market review for safety and efficacy. Merely the processors must follow guidelines in how they procure the donor tissue, how they process it, how they distribute it, and also the keeping of documents of what patient it goes into, tracing all the way back to the original donor. Our next category now is actually a subset of allograft bone, known as demineralized bone matrix, or DBM. I think a discussion of DBM is really best done by first talking about and understanding how DBMs are manufactured, because I think that gives us a lot of insight into their potential mechanism of action and their usefulness when brought into clinical practice. In this slide that I borrowed from University College of London demonstrates very clearly how DBMs are manufactured. The manufacturers start with human cortical allograft bone, and it's then digested in either hydrochloric or nitric acid. As shown in this slide, these investigators took a piece of cortical bone and suspended it halfway into a beaker full of hydrochloric acid. What you see happens here is the acid dissolves all of the mineral component, the calcium phosphate in the bone, which is the portion that would render that bone osteoconductive and allow bone to grow onto it. What's remaining is a mixture of proteins that is between 95 and 98% collagen type 1. Among the remaining non-collagenous proteins include such things as the BMPs, or bone morphogenetic proteins. So in theory, those trace amounts of bone morphogenetic proteins have the potential to render this material osteoinductive. In fact, most of these materials, when they are tested prior to being brought into commercial use, are placed into an athymic rat model, either in a muscle pouch or subcutaneously, and the demonstration of bone being formed in that model is the basis for the marketing of most of these materials. Bay and his collaborators published a pretty interesting study in which they measured directly the amount of BMP2 in a wide number of commercially available DBM products. They measured this by use of an ELISA assay, and the results are summarized in this chart. A couple of interesting things can be found. First, looking from top to bottom, brand to brand, you can see a fairly wide variability in the amount of BMP in these products. As interesting, or perhaps more interesting, they took this a step further and actually opened 3 separate boxes, 3 lots, presumably 3 different donors in each brand. And looking left to right, you can actually see more variability from box to box than there was in brand to brand. In my mind, possibly the most interesting finding from this study is if you look at the units, the amounts of BMP they measured in each of these products was on the order of picograms per milligram, or on the order of 1/1000th of the concentration that is bioavailable in normal physiologic bone healing. Now, this study was then followed up by this study done by Jeff Wong and his collaborators. They did a rat posterolateral spinal fusion model using a number of the most commercially successful DBMs at the time. And interestingly, they found with one of these products, 0 of the animals achieved a successful posterolateral fusion, while at the time, not only was that one of the DBMs shown to have roughly median concentration of BMP as the others, it was in fact one of the most commercially successful products at the time. Subsequent to that, that company went on and effectively failed and went through Chapter 11. That technology was acquired by another company, it was rebranded, and that exact same product is now once again a commercially successful DBM in the United States. So enough about animal studies. I think most importantly is the human clinical experience, and particularly what's been published in the literature. Now, DBMs have been commercially available in the United States for over 2 decades now, so most folks assume that there should be a large number of peer-reviewed published clinical trials. In fact, that turns out to not actually be the case. While there are a number of published clinical studies, to the best of my knowledge, only one of those is a level 1 randomized trial. In that trial, the conclusions were that the DBM was found to be useful in posterolateral spinal fusion. I think it's important, though, to understand, and if you read the materials and methods in that study, it was in fact a prospective randomized trial conducted at multiple clinical centers. Unfortunately, the control group, which was Autograft, was an appropriate control, but the test arm was Autograft with the addition of the DBM. And again, if you read more carefully into the materials and methods, you will find that the average patient in the control group received 20 cc's of Autograft. The patients in the test group received, on average, 20 cc's of Autograft plus the addition of the DBM. So while this has been reported as a prospective level 1 study demonstrating the efficacy of DBM, I think if you understand how this study was conducted, it actually doesn't show that at all. It perhaps merely only shows that the DBM did not degrade the performance of Autograft that was already being used. To date, unfortunately, that is the best clinical study available to support the use of DBMs in spinal fusion. And furthermore, while these materials are marketed with the purpose of being osteoinductive and demonstrated to be so in athymic rats, to date there has been no published evidence demonstrating these materials to be osteoinductive in a human being. Our next category here is perhaps one of the most interesting. We're now going to talk about BMP, and in particular, InFuse recombinant human BMP2. Now, in order to really kick off this discussion, it's always worth taking a step back and looking at the history of how we got to where we are in our current understanding of bone grafting and really the initiation of the development of this category. And that starts with a review of this gentleman, Marshall Urist. Marshall Urist was both an orthopedic surgeon as well as a basic scientist who spent the vast majority of his career at UCLA. His lifelong research interest was trying to understand and unravel osteoporosis to hopefully come up with some solutions and cures for it, but along the way he did some really fantastic and amazing work. One of the things that he did was he was starting to unravel and sort of take apart bone and understand how it works. And he took bone, demineralized it by acid, as we described in the discussion about DBM, and when he placed that into a muscle pouch in an athymic rat, he found that it formed bone in spite of the fact that in that soft tissue site, bone should not form. He termed that bone formation through autoinduction in his seminal paper in Science in 1965, and that was later shortened to be referred to as osteoinductivity. He took that work a little bit further, and using the methods available at the time, the Western blot assay, he found that there was a certain molecular weight range of proteins responsible for that effect. And what I now believe is a huge historical mistake, he named those proteins the bone morphogenetic proteins. But that kicked off decades and billions of dollars of research spent on the development and commercialization both of InFuse, BMP2, as well as the commercial DBMs, which Urist himself never believed had any commercial or clinical value based on the work that they were doing in their labs and in their clinics at UCLA. With the understanding of the ubiquitous biochemical nature of BMP2 and its being involved in so many processes through fetal development as well as in adult life, also understanding the fact that the commercial dose demonstrated to be efficacious in the IDE clinical trial is on the order of 1 million times the physiologic concentration involved in native bone formation, it shouldn't be a terrible surprise that there are sometimes complications unexpected outside of just simple bone formation and healing. Some of those that have been reported in the literature as well as at major meetings include ectopic bone formation, osteoclast hyperactivity, in other words, the turning on of bone resorption prior to bone formation, late-stage radiculitis, rather than seeing radiculitis a couple of days post-op, merely from nerve retraction during surgery, etc. This occurs 6 to 8 weeks post-op, and no one really quite understands why this is happening yet, as well as a number of other things that are well seen in the literature. That being said, this product has definitely been demonstrated safe and effective within its approved indication, which is single-level standalone ALIF. One just really needs to understand that this is not simply a device, but rather a very powerful drug-device combination, and the drug component is a supraphysiologic concentration and does many things in addition to just simple bone formation. The FDA IDE clinical trial, which resulted in the approval of InFuse, was a prospective randomized study in ALIF in threaded titanium lordotic cages. In this study, InFuse was compared prospectively to iliac crest autograft. The primary outcome measure was fusion, as measured radiographically, but also included a number of functional as well as secondary outcomes. In this study, InFuse was shown to have a 93% fusion rate versus 88% with Autograft. It's important to understand that this was a non-inferiority trial and was in fact found to be statistically non-inferior. So while there was a trend to a higher fusion rate with InFuse, that was not a statistically significant trend. The primary outcome, or the only outcome where there was shown a statistical difference, was in the reduction, or rather the elimination, of iliac crest pain at the harvest site with the use of InFuse. The next category I'd like to discuss are the synthetic bone grafts. Now, this is the most numerous category of products commercially marketed in the United States, with well above 200 products currently cleared through the 510(k) process by the FDA. So this category of products encompasses a number of different chemistries, structures, delivery form factors, etc. And I think it's worth taking a minute to talk first about the chemistry and how that relates to its biocompatibility and more particularly its resorption rate. Now, there are only a few chemical compounds that are appropriate for long-term bone implantation. And I've arranged them on this slide in a sort of genericized order from fastest resorbing on the left to slowest resorbing on the right. So if we start and work from the left, we'll begin with the calcium sulfate materials. Calcium sulfate, otherwise known as plaster of Paris. And there are a number of these products that are being marketed. Not as many of them today in spine as there once were. The ones that are still commercially being marketed are mostly focused around trauma and extremities. And the reason for that is that calcium sulfate plaster of Paris is water soluble. So when these materials are implanted, they tend to resorb fairly rapidly and are removed from the implant site, in some instances leaving no benefit in terms of bone formation. Moving to the right along this scale, we next have a group of materials which are collagen calcium phosphate composites, wherein the primary constituent is collagen. Now, collagen itself is not osteoconductive and really will not provide a surface for bone formation, and collagen is resorbed fairly rapidly through enzymatic digestion. So these materials are generally understood to resorb too quickly for the majority of bone formation applications, and particular those in spine, which tend to be fairly challenging. If we now jump all the way to the right end of the spectrum, we will find the materials such as synthetically derived synthetic crystalline hydroxyapatite. Now, while the normal bone mineral is in fact crystalline hydroxyapatite, it is of an enormously different scale, being on the order of nanometer scale crystals. It also has chemical defects in it such that it can in fact be resorbed, but synthetic crystalline HA is effectively non-resorbable in any clinically relevant time frame. A number of companies have prepared composites of hydroxyapatite and other calcium phosphates such as TCP. These may resorb faster than HA, but in practice, these are largely non-resorbable as well. In the middle are materials that have been found to resorb in clinically relevant timeframes, and those are generally tricalcium phosphate, as well as anorganic bone mineral derived from native bone, but retaining a number of crystallographic and chemical defects to render it resorbable in a reasonable time frame. The next category is really fun and interesting to talk about, and these are the stem cell products. So the stem cells are a subcategory of the HCTP human tissue products, and these products are being marketed on the basis of having large numbers of stem cells from the donor that have a biological function leading to bone formation when implanted into a patient. Now, the processes by which these are procured and manufactured are all different from brand to brand. The claimed number of retained cells are different from brand to brand. But fundamentally, there are some real significant challenges with the whole concept here. One of which is, what cells are being delivered exactly? What is the function of those cells when they're implanted? What are they actually doing? And what is the demonstrated efficacy in terms of bone formation, and in our instance of what we're talking about here, spinal fusion? Because these are non-regulated HCTP products, there is no requirement for demonstration of safety and efficacy. Because these are human cells, they really can't be tested in normal animal models because of the histoincompatibility. The only way they could be tested in animal models is in athymic rats or other immunologically privileged or compromised animals, and as such, those are expensive models to do and are typically not done. So one of the claims that leads to the safety of this category of products is that mesenchymal stem cells, which are presumably the majority or some of the cells that are being transplanted, are immunologically privileged. What does that mean? An immunologically privileged cell is one that does not have surface antigens of the donor cadaver from which they came, and therefore should not be recognized or attacked by the immune system. In this rather interesting paper by Liu et al., they looked at the immunologic privilege of mesenchymal cells and found that they are, in fact, immunologically privileged. They will not be recognized as invaders when implanted. One of the challenges and problems with that, however, is that for these cells to function, and the idea, at least initially, was that they are going to differentiate down the lineage of forming osteoblasts, and they're going to, in fact, themselves participate in the bone formation process. Should these mesenchymal cells, in fact, differentiate down that or any other cell lineage, they will then express the surface antigens of their donor. They will then be recognized as foreign invader cells and they will trigger an immune response. Most likely they will be destroyed by T cells of the recipient, the recipient, and lead to potential complications associated with that immunological response. We don't really understand what is happening when these cells are implanted. We certainly don't understand what the efficacy is, and we don't understand what the safety is should these cells remain alive, as is claimed, and then remain alive inside of the recipient as well. While these stem cell products have now been marketed in the United States for well over a decade, there is surprisingly little published in the clinical literature to support their use. That in spite of the fact that on clinicaltrials.gov there have been a number of clinical studies ongoing for most of that time. To date, there only have been 2 studies published, and both of those very recently. One of those was what appears to be a small cohort from a much larger study, 300 patients on clinicaltrials.gov, 33 patients in the publication. This was a prospective, non-randomized study that showed positive results, but only using plain film to evaluate fusion. This second study is a randomized trial conducted by people not sponsored by the manufacturer. This was an ACDF study and actually found inferior results in fusion with the stem cell product as compared to Cancellous Allograft. So to review the stem cell category, certainly the idea of taking a large number of stem cells from a donor, placing them into the patient to drive spinal fusion is a very compelling one. With the products that are currently commercially available, starting from recently deceased cadaver bone, removing the remaining viable cells, changing those or eliminating the cells that we don't want to end up with a subset of cells, cryogenically preserving them, thawing them and implanting them into the patient, really doesn't seem to be fulfilling that promise. So reminder, these are HCTP products, so there's no review for safety and efficacy from the FDA. To date, there is not compelling clinical evidence to support their use in the published literature. And if these cells are MSCs when implanted, and they should differentiate down the pathway towards becoming osteoblasts with the potential to form bone, they will express the surface antigens of their donor, and they will be destroyed by the patient's immune system, perhaps triggering some sort of a local immune response. So to date, spending almost the cost of BMP to use one of these products on the basis of the currently available evidence doesn't seem to be supported at this time. I'd now like to take the opportunity to introduce a brand new category of bone grafts, peptide enhanced. Now, to date, there's one product available in this category, but there are other peptides currently under development and investigation. P15 is a 15 amino acid peptide, which is a naturally occurring segment of the human type 1 collagen molecule. If you look in all human connective tissues, the cells that work throughout those tissues and develop and remodel those tissues all use this same attachment point in collagen type 1, where they attach and they actually use it for motility to move throughout those tissues. If you take and pull out and synthetically manufacture this 15 amino acid peptide to be identical to the natively occurring one, and you coat a surface of a material with this peptide, connective tissue cells of all types will attach very, very aggressively all over the surface of that material. What we do is we have synthetic P15 manufactured for us, and we then combine that with an anorganic bone mineral. We coat the P15 peptide onto the surface of the ABM, and that's then placed in a carboxymethylcellulose carrier. When that is implanted, the P15 in there will attract, attach, and activate bone-forming cells from the surrounding tissues. This is a unique mechanism of action not found with any other category of bone graft materials, and this unique mechanism of action results in very powerful bone formation. However, it retains a very safe profile with very little opportunity for side effects. This unique mechanism of action ensures the safety of this technology when it's implanted. Now, by virtue of this attract, attach, activate mechanism of action. It is simply taking advantage of and amplifying the native biology already available in the site and in the patient. iFactor peptide-enhanced bone graft has a long-standing and successful clinical track record. This track record began with the core P15 ABM technology being brought into use in dentistry in the 1990s, and in these applications has been used in over 500,000 patients to date. The application and use in spine and orthopedics began with the CE mark in 2008, and subsequently in November of 2015 with receipt of FDA PMA approval for use in anterior cervical discectomy and fusion. This PMA approval was on the basis of an IDE, prospective randomized clinical trial, the results of which have been published in Spine. The IDE clinical trial, which resulted in the approval of iFactor, was a prospective randomized trial of iFactor versus local autograft inside an allograft ring on a single level fusion with an anterior plate. In this 319 patient study was 1-to-1 randomization, iFactor versus Autograft, with 4 primary outcome measures. Those were radiographic fusion, neck disability index, neurological outcomes, and safety. All patients were being followed to 6 years, and the primary outcome was measured for FDA submission at 12 months. This trial had a non-inferiority statistical design, and you'll see all 4 of the primary outcome measures in this slide showed non-inferiority to Autograft. All of them had slightly better results with iFactor versus Autograft, but they were not statistically different. In the prospective statistical design for this study, we included an overall success metric. This overall success is a measurement of individual patients who were successful for all 4 of the primary outcomes. And what we found at 1 year at the time of FDA submission was not only a significantly higher number of patients successful with iFactor versus Autograft, but that in fact was statistically significant as well as clinically relevant. We then followed and recently collected the 2-year data. We again showed at 2 years non-inferiority of all of the primary outcomes as anticipated, but the statistical superiority in overall outcome versus Autograft was retained at 2 years as was shown at 1 year. Spinal fusion in the United States is largely done on patients with private insurance. All of the major payers today have published coverage policies whereby they describe which patients are eligible for spinal fusion and reimbursement, as well as going into some additional detail. One of the interesting features of these coverage policies is that the majority of payers list bone graft technologies, categories, in some instances individual products that are considered to be on policy and therefore subject to reimbursement. It's very interesting to note if you go and read these policies, which are mostly available online, you will find almost all policies for cervical fusion list Autograft and Allograft as being covered, but they generally list DBM, BMP, synthetics, and stem cells as being not covered. What does that mean? What that means is that they're not paying for these products individually. They're paying for the entire procedure under a DRG. However, they are reserving the right to potentially reject the DRG payment for the procedure, either in pre-certification or in more rare instances after the procedure has already been performed. So I'd like to conclude the discussion of bone grafting categories with a review of which of these technologies or categories can be supported for use in spine on the basis of peer-reviewed level 1 clinical data. I think it's easiest to do this by putting these into buckets. The first of which is those that can clearly be supported for use in spine on the basis of published level 1 data. Those are Autograft, InFuse, and iFactor. The second group are those that have level 1 published evidence, but with some caveats. The first of these is Allograft Cancellous chips. Now, the literature does support the use of Cancellous chips as an autograft extender or by itself in some low-demand applications such as adolescent scoliosis. The second of which is Vitas, and the level 1 data supporting its use in spine is only when used in conjunction with iliac crest autologous bone marrow. Unfortunately, that leaves all the remaining products without quality level 1 data in the literature to support their use. Those being the synthetic bone grafts, excluding BTOS, the DBMs, and the stem cell products that have come into fairly wide use. I'd like to thank you for your attention. If you wouldn't mind, please let us know if there's anything that we can do to follow up or perhaps answer any of your questions.